Today we're talking about Chapter 5, Section 1, which is all about the cell cycle. This is going to be the generalized life cycle of a cell, and within this discussion we will also talk about the reason why cells tend to be small. It has to do with the size of the cell and its ability to get the nutrients that it needs, and that is linked to how often a cell is going to divide. So let's begin by talking about the four main stages of the cell cycle. 3 of these stages are going to be a part of what we call interphase here. So interphase contains the gap 1 stage. That's going to be mostly just growth and normal functioning of a cell. We have the synthesis stage. Synthesis refers to making the DNA. This is when DNA replication occurs. And then we have another stage of additional growth and preparation for division. The 4th main phase of the cell cycle is this slice over here. Notice this is called the mitosis or M phase of the cell cycle. And the M phase is going to include all 4 stages of mitosis itself. Mitosis is technically the division of just the nucleus of the cell. And then that is followed by cytokinesis, which is the division of the cytoplasm of the cell. But all of those together make up what we call the M phase of the cell cycle. So to abbreviate the stages of the cell cycle, we have G1, then that stands for gap 1. We have the S phase for the synthesis of DNA. We have G2, which is that additional growth and preparation for division. And then we have the M phase, which is when mitosis and cytokinesis are going to occur. So let's look at those in a little bit more detail. I introduced them on the last slide, but let's look at the actual detail of each stage. You'll want to make sure that you have this noted on your sheet. You have this diagram on the Power Notes sheet. So make sure that your labels and the descriptions match up to the diagram on the sheet. So we're starting out, if we think about the beginning of our cell cycle, this is kind of the starting point right here. So a cell that has just divided, a brand new cell, is going to start at this point, and it is going to begin interphase. Interphase is by far the longest phase of the cell cycle. Cells are going to spend most of their life in interphase, probably within the gap 1 stage. So this is when the cell is doing its normal thing. If we're talking about a muscle cell, the muscle cell has a lot of mitochondria making ATP so that it can contract the muscles and allow you to move around. If we are talking about a cell in the digestive system, it's probably secreting enzymes to help you digest your food. Or maybe it's part of the intestinal lining to push the food through your digestive system. Whatever a cell normally does, it is doing its normal function here in the gap 1 phase. Now, if a cell is going to divide, and we will talk a little bit later about those cues for cell division, but if it is destined to divide, it is then going to enter what we call the S phase, and the only purpose of the S phase is to copy the DNA. So this is going to be a process that's very focused on what's happening in the nucleus. We're not really doing a lot of the other functions of a cell. A cell is going to stop doing its normal function because it's going to be messing with the DNA, and it's hard to use your DNA to make essential proteins if you're in the process of copying it. So the S phase really only has one thing that's happening, and that is the process of DNA replication. After the DNA has been copied, there's one additional growth phase. A cell will continue dividing, making sure that it has enough organelles and has all the proteins that it needs to undergo the process of mitosis. So then when mitosis begins, again, the M phase right here includes all of mitosis and cytokinesis. Mitosis is the division of just the nucleus of the cell. So we're talking about eukaryotic cells here, not prokaryotes. They do not go through this process when they divide. It's much simpler for a prokaryotic cell. And then after the nucleus divides, We have to split the cytoplasm between the two new cells, and that is called cytokinesis. So we are talking a little bit later about regulating the cell cycle. And it's important to note that this process of mitosis, so at this point right here in our cell cycle, a cell is not going to go from G2 into mitosis unless it is a large enough size and the DNA has been copied and checked to make sure that it is error-free. So now let's talk about the rates that cells divide. You do not need to memorize this chart, but it is interesting to look at the different types of cells in the body and their approximate lifespan. Because the rate of cell division for different cells in the body, it's not always the same. It depends on what that cell is normally doing. If the cell gets worn out, for example, like your skin cells, these cells on the outermost part of your skin are constantly being shed. It's part of the function of the skin. Something like the intestine, notice that they only live about 4 to 5 days because, again, they're lining the tissue of the intestine. They have a job there. They get damaged. They get scraped off by the food that's moving through your intestines, so they need to be replaced pretty quickly. But something like a liver cell, it lasts much longer. A red blood cell lasting 4 months. So some of these cells that don't get as much wear and tear, they're going to last much longer. And some cells that aren't even listed on here, like your nerve cells in your brain, they live the entire lifespan of the organism. They don't ever divide. So however old you are, that is how old your nerve cells are. So speaking of those nerve cells, because they perform such an important role, it's too risky for the body to kind of take them out of commission and try to divide and copy those cells. So any cell that is not currently preparing to divide, instead of going through the cell cycle, so this would be our interphase here with G1, S, G2, and then there's mitosis. In G1, if a cell, like a nerve cell, is not going to further go on to copy its DNA or go through mitosis, it enters what we call the G0 phase. It might stay there permanently. A nerve cell would stay there permanently in the G0 phase. It's functioning, it's doing its normal thing, but it's never preparing to divide. Other cells, like those liver cells that we talked about that can last for 500 days, they would enter the G0 phase for 499 of those days, and then when it's time for them to divide, they would exit G0, come back into the rest of the cell cycle, copy their DNA, grow, and then divide here in mitosis. So what we're focusing on here at the beginning of the unit is how cell size can cause a cell to divide. What you're looking at in this picture are stained onion cells. The onion is kind of the part of the root of the plant. The roots come from the onion and they grow very quickly. So it's very common to see onion cells in various stages of division. You can tell that there are some larger cells here that are just beginning their division. These cells down here have just finished their division with the new cell wall in between them. They're much smaller. And it's really an increase of size that's the main cue for these cells. Once these cells grow as a part of the root and they get larger and larger, eventually they'll reach a size where it is time for them to divide because they are not going to be efficient at obtaining nutrients anymore at that large size. So the limits on cell size, there's really 3 things to keep in mind when we're looking at the size of different cells. The first thing to keep in mind is that volume increases faster than surface area. We will be talking about both volume and surface area here. The surface area of a cell is whatever is on the outside. Basically, it's the amount of membrane that a cell has. Versus the volume is going to be the space that that cell takes up. So think of the volume as being the cytoplasm of the cell. And what a cell needs, as we will discuss, and we'll do a lab on this as well, A cell actually needs to have a large enough surface area or enough membrane to be able to support its entire volume. So we're going to start to look at something called the surface area to volume ratio. This first cell here has a 6-to-1 ratio of surface area to volume. It's easy to calculate surface area and volume here because we are assuming these are cells that are cube shaped, so we can just use the equations and formulas that you've learned in math to calculate both surface area and volume. But when you divide it out and you get the ratio, we can see we have a 6-to-1 ratio of surface area to volume in the small cell, but a 2-to-1 ratio of surface area to volume in the large cell. Now remember, it's the cell membrane that allows things to pass in and out of the cell. So a large ratio here, 6-to-1, means there's 6 times the amount of membrane relative to the volume, versus in this big cell here, there's only 2 times the amount of membrane as there is volume in that cell. So in general cell biology, you find that cells must be small. The smaller the cell, generally speaking, it's going to have a larger surface area to volume ratio. So the other two things to consider about the cell size limitations is, first of all, this surface area to volume ratio needs to be large. We just kind of discussed that, how a small cell is going to be more efficient at exchanging nutrients. That's because it has a large surface area to volume ratio. So there's an inverse relationship there. The larger the cell, the smaller its surface area to volume ratio. And a large surface area to volume ratio is preferable, and that's why we tend to see cells that are very small. Now, sometimes a cell has to be large in order to do its job, but it still needs to be able to have a large surface area to volume ratio. So there's a few ways that that can happen. Changing the shape of the cell, flattening it out, like these blood cells here are very flat and disc-shaped. This would be a nerve cell over here. It has these really long sections that stick out. A nerve cell can be a really large cell. There is a nerve that runs from the base of your spine all the way to your big toe. One continuous cell. Obviously, that's going to be a fairly large cell. So in order to have a large enough surface area to volume ratio, it needs to have all of these elongated portions to really maximize the amount of surface area on that cell. So that's the basic information about the cell cycle and how it relates to cell size. You also have on the back of your note sheet a copy of a POGIL, and it's just Model 2 of this POGIL, where you're going to have an opportunity to compare different sizes and shapes of cells and answer some questions here to practice with that information. So go ahead and take some time to work on that. If you have any questions, as always, please make sure you ask them when you come to class. And hopefully you found this helpful.